Hello, I'm solving paper 4 biology, specimen paper 2022, 9700 variant 4 today. So the syllabus changed. C4 plants were excluded from photosynthesis. The menstrual cycle, 6 hormones, these were also removed. And a small part was added in genetic engineering. CRISPR and Cas9 were added, gene editing basically. These have been added to the syllabus from 2022 onwards. Moving on, let's start. The II is a mammal native to Madagascar. IIs are active at night and make their nests high up in trees. They feed on insect larvae. In the trunks of trees. Figure 1.1 shows an II. The IUCN categorizes the II as endangered. This means that the II has a very high risk of becoming extinct in the wild. Name the domain to which the II belongs. So, taxonomy, right? So, what are the levels of taxonomy? At the top, we have domain, followed by kingdom, followed by phylum, class, order, family, genus, species. This is all we need to know. We just need to know the order. That's all. So we need to know about three domains. There are three domains: eukarya, bacteria, and archaea. Eukarya are eukaryotes, and archaea and bacteria are prokaryotes. So we need to know a few kingdoms that are under the domain eukarya: Plantae, Animalia, Fungi, and Protoctista. That's all we need to memorize. We don't need to remember anything else. Okay. So II is clearly they want the domain. So we have three options for domains, right? We have eukarya, bacteria, and archaea. So clearly, since it's an animal, they are part of the animal kingdom, right? It is part of the animal kingdom, so it's going to be eukarya. State one reason why IIs may have become endangered. This is a common question. Typically, this is due to habitat destruction. Habitat destruction. Habitat, you could also say habitat fragmentation or deforestation, or you could classify it as logging. What are alternate points though? They may have suffered from disease, or this may be due to hunting, or any alternate valid point works, like a genetic diversity, low genetic diversity, or large range and low population density. Large range basically means a long distance between them, so there would be no gene flow, right? Large range and low population density. Okay, so suggest ways in which zoos may be able to protect this species from extinction. So how may zoos help? This is really important. They may help in captive breeding. This would then lead to releasing the IIs into the wild, right? They could release the IIs into the wild. Or zoos could help in building conservation projects in Madagascar, or establish some reserves. Okay. Zoos also help in raising awareness or providing education. Raising awareness or providing education. They could also research, feeding habits or their behavior. Last but not least, there are vets present in zoos, right? So they could provide medical care. So it's actually veterinary care. Provision of veterinary care by zoos. They could also help in raising funds, raising funds for a certain animal which is endangered. So there are two main island populations on the island of Madagascar, one in the west and one in the east. Figure 1.2 is a map of Madagascar showing the location of the two main populations. This is a map of Madagascar showing the location of the two main populations. A study into the variation in the DNA nucleotide sequence of IIs showed that there is a large genetic difference between the west population and east population. The two populations of IIs may be evolving into separate species. Basically, speciation may be occurring. Why may that be possible? Due to a river in between these two sections, and also due to this, mountain range. So maybe there is no gene flow between the east and west side because you would have to cross this mountain to mate with someone from that side, right? So it might be very unlikely, which leads to no gene flow. And there may be different selection pressures on either side of the mountain range, right? As a result, this actually leads to different selection pressures, and different alleles will have an advantage, a selective advantage, which ultimately changes the allele frequency over time. That's what happens. So, with reference to figure 1.2 suggests why there is a large genetic difference between the two populations. It's pretty straightforward. This is geographical isolation, right? Allopatric speciation. So, mainly geographical isolation or barrier exists between the two species. You could give an example. Due to the river, right? Due to the rivers or the mountain range, okay. So you could say that the west and east population are unable to interbreed, or you could say that there is no gene flow between the populations, okay. Also, there are different selection pressures or environmental factors acting on west and east populations. So different alleles are selected for. So this is a typical question on geographical isolation. Okay, you will follow this marking scheme all the time. This is a typical scheme for geographical selection. So you're going to say that there's no gene flow between the two populations. There are different selection pressures acting on both of them, right? This actually leads to different mutations. What is a mutation? A mutation is a spontaneous change in the base sequence, right? So there are different mutations in west and east populations, right? As a result, different alleles are selected for. Okay, so different alleles are selected for. So you could refer to genetic drift. What happens in genetic drift? Basically, when suppose the east population was not there initially, somehow a few of these animals made it across the mountain and went to the west side. Okay, this is a new population. So when you have a very small gene pool from the original population that goes to another part of the island, they're going to experience different selection pressures, and that effect will be more pronounced on smaller populations. Okay, there was a question like this in the February March 2022 variant, where a small proportion of the recessive population was taken to a small island, and it was seen that the recessive population became the, you know, dominant part of the main population. Okay, so that's genetic drift. Genetic drift may have occurred. Right, genetic drift may have occurred. What else? They may have been separated for a long period of time, right? West and east population may have been separated for a long period of time. Right. So this actually leads to a change in the gene pool. Name the type of speciation occurring. I was telling you guys this is allopatric speciation. So we're seeing allopatric speciation here. They gave sympatric speciation in the uh, February March 2022 variant. Really important for the May June series. Starting question number 2 now. Figure 2.1 shows a diagram of a motor neuron nerve cell. Basically, name the structures labeled A, B, and C. So A is basically a gap between the myelin sheath, right? So it is actually a node of Ranvier. What about B? B is not the gap, it's actually what's in between. It's part of the nerve cell. So a nerve cell has two types of projections, right? One that carries the impulse away from the body, and one that brings impulses towards the body. So these ones are the dendrites that bring impulses towards the nucleus or the body. The one that takes the impulse away, that's called the axon, right? What about C? What is that part called? It's called the cell body. You could also say soma, but it is not recommended. So what is the function of a motor neuron? Basically what it does is it transmits impulses or action potentials from the CNS or from sensory neurons in the case of, reflex actions, or relay neurons, or even their synonym, intermediate neurons, right? To what exactly? To effectors, muscles, or glands. Any one of these will work. Okay. With reference to figure 2.1, explain the fast transmission of impulses along motor neuron. They're basically talking about saltatory conduction. Okay, they're talking about saltatory conduction. So let's describe this. This is transmission in a myelinated neuron, which increases speed. So Schwann cells wrap around the axon, or you could just go for B since it's on the diagram, to form the myelin sheath, or you could just say that the axon becomes myelinated. So what does this myelin sheath do? The myelin sheath actually insulates the axon. The myelin sheath insulates the axon, or you could just refer to there being a lack of movement of ions. The myelin sheath causes a lack of movement of ions, but does it completely stop the movement of ions? Not really. it can only occur in some places. Okay, where can the movement of ions occur? So the depolarization, or you could just go for the action potentials, can only occur at nodes of Ranvier, or you could just refer to A. So what actually happens? A local circuit is set up between the nodes of Ranvier, right? So what does this local circuit help to do? The action potentials can actually jump from one node to another, right? Action potentials jump from node to node, and this phenomenon is actually called saltatory conduction, right? So you could say that this actually, like, since it's mentioned in the question, we can't actually talk about it. But typically, there's another marking point. You could say that this causes fast transmission of impulses along the motor neuron if it wasn't mentioned in the question. Okay, really common question. I've seen this in the winter session too, 2018, October, November. So remember this. Moving on, corals grow in shallow seawater. This is question number 3, right? Corals grow in shallow seawater. Corals consist of colonies of small animals called polyps. These polyps have photosynthetic protocysts called algae within their cells, which is advantageous both to the coral polyps and to the algae. The algae that live within the cells of coral polyps can also live independently as free-living algae. The rate of photosynthesis of algae that live within the cells of coral polyps is higher than that of free-living algae. Suggest and explain why the rate of photosynthesis in algae that live within the cells of coral polyps is higher than that of free-living algae. Okay, so this is a suggest question and explain. So we could talk about multiple points really. There's room for error. So maybe since there are more, right? More algae together. Suppose there's only one here and there are more living together in polyps, right? So they're going to respire more, right? So we could say that the carbon dioxide CO2 concentration is higher or increased, right, in the polyp. So CO2 concentration is no longer a limiting factor. So CO2 concentration is no longer a limiting factor. So where is this carbon dioxide coming from? You could say that the CO2 is from coral or polyp respiration. So this increased CO2, right, this increased carbon dioxide can be used for the Calvin cycle or light independent reactions in algae. You could talk about a further detail, like what? You could say that since there's increased CO2 concentration, what can be used for? CO2 is fixed onto RUBP, right? Using Rubisco, or you could say it binds to RUBP. Same wording, it's fine. Different wording, but you're gonna get the mark. So this is one route. You could answer it this way by talking about carbon dioxide concentration. What is the other route? You could also opt for this. This is your secondary route. You could go for the temperature, right? Temperature is higher. the temperature is higher or increased. So the temperature is no longer a limiting factor. So the increased temperature, where does it come from? Is due to the coral or polyp respiration, right? Now, basically we have the same points, right? Just the last point is a bit different. So the increased carbon, it's going to be the increased temperature. The increased temperature is for the the Calvin cycle or the light independent reactions in algae, right? So what is the effect on this actually? So temperature is always linked to enzyme activity, right? So higher or increased temp leads to greater activity of enzymes, Basically, we can talk about Rubisco, or you could say that it leads to greater rate of enzyme catalyzed reactions. Greater rate of enzyme catalyzed reactions. Okay, that is all. Moving on to B, the algae that live within the cells of coral polyps have 5 different chloroplast pigments. Table 3.1 shows the light wavelengths at which each algal chloroplast pigment shows its 2 largest peaks of light absorption. So, corals can be kept in glass tanks that are usually lit by lamps radiating mainly violet and blue light with wavelengths in the range 400 to 490 nanometers. With reference to table 3.1 suggests why lamps radiating mainly violet and blue light are expected to increase the growth of coral polyps more than lamps radiating light of all wavelengths. Okay. So guys, think about the visible light spectrum. It's VI, B, G, Y, or VIBGYOR, right? So it's in the order of increasing wavelength from around 400 nanometers to 700 nanometers, right? So clearly, um, corals can be kept in glass tanks that are usually lit by lamps radiating many violet and blue light with wavelengths in the range 400 to 490. So as you can see, they mainly absorb radiation from 400 to 490, right? Even here, it's between 400 and 490 almost. This is an anomaly. This is also an anomaly. You could say that. So using all colors would be a waste because the corals or the polyps do not absorb those wavelengths, right? So we need to use wavelengths in this range of 400 to 490 nanometers, basically. So how to write this answer? Let's learn the wording. So pigments absorb violet, blue, or 400 to 490 nanometers, or the lamp colors will, or best, or most, or you could just go for probability at 8 out of 10 peaks, right? So the rate of photosynthesis of algae increases with more light absorbed. So how does this even relate to growth? Like if there's more photosynthesis, where is this energy used? Basically, if we have more algal photosynthesis, right, it can produce more food. That food can be used for the coral in general because there is a transfer of organic nutrients or oxygen two polyps from algae. Think about it. What does photosynthesis do? It mainly takes in carbon dioxide and produces oxygen as a byproduct and food. So that oxygen can be used by the coral, right? It can be used by the coral and it can use that for growth or respiration, right? So it's not like the products that the algae produce, right, are used by the coral. It's just that since more photosynthesis is occurring, the oxygen released the oxygen released is used by the coral for its own needs. Okay, so we can just refer to this. There is a transfer of, you could say, organic nutrients. That's fine, they accept this, or oxygen, to polyps from algae. Now, why does coral growth increase actually? Coral growth increases with more algal photosynthesis. There's an alternate valid point. Basically, the where the coral lives, right? The basically violet blue light is more common there at the depth where they're at currently, right? This is an alternate valid point because think about it. Plants or algae are adapted to where they live. Why would it absorb these properly? Because they always live there underwater, right? So obviously there's an adaptation. There was a selective advantage for those who could absorb these wavelengths, right? So this is the last point. Violet, blue, or 400 to 490 nanometer light is the same as where the corals live. Okay, that's all. Photosynthesis is in the algae living within the cells of coral polyps. It's the same as photosynthesis in plant cells. Outline the process of cyclic photophosphorylation. Okay, so this could come in the form of a broad question as well, so you should learn that. It's pretty common. So we're gonna write a few points. So cyclic photophosphorylation only involves photosystem 1. Okay, so light energy is absorbed by accessory pigments. So it's like this, right? It's set up like this in a funnel shape. So the accessory pigments are over here and chlorophyll A is at the reaction center. So mainly the light energy is absorbed by the accessory pigments. They pass the energy onto the reaction center. So this energy is passed onto chlorophyll A, right? Or you could say that it's passed onto the primary pigment or the reaction center. Anything works. Okay, so what is this energy used for? It is actually used to excite an electron, right? An electron is excited to a higher energy level, or you could just say that the excited electron is emitted from chlorophyll A. Now where does this go, right? So this was our photosystem 1. It gains some energy, it's excited. Now where does it go? It actually passes along a few electron carriers, the electron transfer chain, right? So the electron is accepted or collected by an electron acceptor. It is passed along the electron transport chain. Right, so it passes along the EDC, and think of it as a ball rolling down a hill. The energy will be released, right? So the electron had some energy. This will be released in the process of doing so, and ATP is produced in the process of doing so by, what's the process called? Chemiosmosis. The same thing happens in oxidative phosphorylation. So at the end, what happens? The electron returns to chlorophyll. Remember that. Or returns to photosystem 1. That works. Okay, everything's fine. These are separate points. So 84 from here would work. Okay, you'll get full marks. Moving on to question 4 now. The PCR polymerase chain reaction is used to produce large quantities of DNA from a very small original sample. The main steps of one PCR method are shown in figure 4.1. What is step 1? They heat the mixture of DNA, which is called denaturation. Okay, why is it done? Mainly to separate the strands. All right. Then step 2 is cool the mixture to 65, incubate it at 72, and then repeat. So what's the second step called? Annealing, and then it's extension. So what's the purpose of step 2? Mainly we attach the primers to the DNA so that DNA polymerase can bind to DNA. It doesn't bind to a single strand. Okay, now DNA replication can occur. And step 3, extension, basically occurs, right? Why do we use stack polymerase? Basically because it's heat stable. It increases efficiency of the process as a whole. So this is quite a common question. So why do we heat it first? It is done to separate the two strands, or you could say that it is done to denature DNA, or you could say it is done to make single-stranded ssDNA, right? How is this actually done? It is done by breaking the hydrogen bonds between bases, right? So that the bases are exposed to produce template strands for complementary copying. So I've written 4 points here, any two would work. So mainly the goal is to make double-stranded DNA, single-stranded DNA, so that the bases are exposed and now we can copy the DNA. It can act as a template strand. So what about primers? What are they? What do we use them for? So primers, or the primer actually binds or anneals to DNA, right? By complementary base pairing. So it doesn't attach anywhere, it attaches to a specific section. So it attaches to a specific, or it attaches close to, okay, it attaches close to a specific section. Section of DNA, right? Why was this done? Mainly so that DNA polymerase attaches to double-stranded DNA, right? Or you could say that the primer. All right. It doesn't bind to single-strand DNA. For the active site, it's the complementary shape is actually double-stranded DNA. So there's an alternate point. You could also refer to forward and reverse primers. So what are these, if you're wondering? Mainly it works in an anti-parallel nature. So there are two strands, right? So one primarily attaches here and the other attaches here. So it goes like this, all right? So that's basically it. Forward and reverse primers. You can just refer to them. You'll get a point for that, okay? So explain why Taq polymerase is used. Why not any other enzyme like the normal DNA polymerase from our body? Why bother using Taq polymerase, right? So the main purpose is it is heat stable, right? Thermus aqueous Taq polymerase is heat stable, or it works at high temperature. Okay, it works at high temperature. Or you could just go for this. You could say that other polymerases are not heat stable. You could say the negative point, right? Like, why not use the other ones? So what's the benefit? So it does not need to be added again for each cycle. It does not need to be added again for each cycle. Or you could just say that it needs replacing only after a number of cycles. Okay, only after a number of cycles. Alright, the negative point exists. Other polymerases need replacing regularly. So we don't have this disadvantage for Taq polymers. Now, what's the end result? The process is more efficient, right? Or faster than DNA polymers, right? So this is actually a question from 2017, I guess. May, June, I'm guessing. Mainly how they make specimen papers. This is the process. They say take some questions from previous years and just make a new paper. That's literally it. If new syllabus content has been added, they just add a few questions. So the presence of a faulty allele of the gene BRCA2 can lead to an increased chance of developing breast cancer. There are many different faulty alleles of the gene BRCA2. BRCA, pronounced BRCA. People who are considered to be at risk of breast cancer may choose to be tested for the presence of these alleles in their genomes. Microarray can be used to test blood samples for the presence of these alleles. The microarray contains DNA probes for different faulty alleles of the BRCA gene. So, what does genome mean? So, what does genome mean for an individual like me? Basically, humans have 23 pairs of chromosomes, 46 chromosomes. So all the chromosomes, right, in my body, all the genetic material in my body, all the combined nuclear and mitochondrial DNA of my body is actually called my genome, the human genome. So it is basically all the DNA, right? All the DNA or genetic material in a person's cell, actually, because all of my cells contain the same thing, right? Except my gametes, obviously. Or you could go for the combined nuclear and mitochondrial DNA. Suggest which type of cell from a blood sample is suitable for testing the presence of these faulty alleles. So no RBC would work, okay, because RBC, red blood cells, do not have nuclei, okay. So it has to be, you could go for a named white blood cell, like you could go for a lymphocyte, okay, or a monocyte, or a macrophage, right, or a neutrophil, or you could just go for a WBC, or you could go for leukocyte. That's a broader term, right? What's the reason? Because it contains nucleus, or it contains DNA. So this was basically a trick question for the weaker students who would go for RBC, unaware that an RBC does not have a nucleus. Okay, so I had this question written. Outline how faulty alleles of the BRCA2 gene can be detected using a microarray. So this is how it works, right? This is the microarray. It has a lot of wells containing probes, short lengths of DNA, which are complementary for the faulty allele, right? So we basically obtain DNA from the individual to be tested. We obtain it from the genome by cutting DNA, or if we want to detect a specific expression, right, we actually get the mRNA for the BRCA gene expression, and using reverse transcriptase, we convert it to cDNA, right? And then we tag the cDNA with fluorescent markers. Then we allow the cDNA to hybridize with the probes on the microarray. Um, if they hybridize, they'll stay. If they don't, we wash off the extra ones, right? And then we view it under UV light to see if fluorescence occurs. The greater the fluorescence, the more the expression of the gene. So how to answer this? There are multiple variations of this question. So first, we can talk about probes or reporters. They're also called reporters. Probes preferred. Probes are short lengths of single-stranded DNA, which are complementary to the faulty alleles or the faulty DNA being tested for. So many copies of one type of probe is placed in each cell of the microarray. So suppose I'm going to place type A over here, type B over here, type C over here, and so on. Okay, many copies of one type of probe is placed in each cell of the microarray. So the sample DNA is made single-stranded. In this mark scheme, they did not talk about how it was obtained, but mainly it's obtained from the mRNA using reverse transcriptase, or you could get it from the genome genes, cut it and make it single-stranded by denaturing it. So the sample DNA is made single-stranded and labeled with fluorescent tags. So it is made single-stranded, then labeled with fluorescent tags. The purpose of making it single-stranded is so that it binds with the probes, right? Because if it was double-stranded, it wouldn't bind. Then we label it with fluorescent tags. Now what? The faulty DNA actually hybridizes or binds with the single-stranded DNA or probes. Let's go for probes. Yeah, probes. Okay, because the probes are already present on the microarray, right? So we are getting the sample DNA from the individual who we think has faulty DNA. It actually hybridizes with the probes on the microarray basically. So the unbound sample is washed off and UV light is used to detect fluorescence. So you could also go for it like this. You could say the bound sample DNA will not be washed off, right? Then laser or UV light is used to detect presence of fluorescence or presence of hybridized probes. So what does the hybridization mean? It means that there is more expression. Hybridization means that there is more expression. That means the individual has faulty alleles, right? But if there's no fluorescence, that means that there is no expression, then the gene is fine, okay? That's the advantage. So outline the advantages of screening for faulty alleles of the BRCA2 gene. So if the disease is not present, it will remove worry. And if the individual gets to know, right, this gene is mainly for breast cancer, right? If the individual gets to know, the woman may change her lifestyle, right? So the informed choice about making lifestyle change, then she may be informed. There will be an informed choice about whether preventative treatment needed or not, right? And if the faulty allele is not present at all, it actually removes a lot of worry, right? It actually removes a lot of worry. What else? This actually allows preventative treatment that may be cheaper than treating the disease itself. So remember, guys and girls, treatment is always, prevention is always better than cure, right? So the earlier you get to know about cancer, the better, or any disease in general. Okay. So it actually allows informed choice about having children. Also, there are some positive implications for life insurance. Life insurance may cover that. Okay, that's all. So I've written about 1, 2, 3, 4, 5, 6 points. So any 4 from these, right? Really important question. So remember to learn this. Moving on to question 5. The contraction of striated muscle is explained by the sliding filament model. Describe what happens in the sarcomere when the myosin head releases ADP and inorganic phosphate. Okay, so mainly remember that when the myosin head binds to the exposed binding site on actin, it causes a power stroke which actually shortens the sarcomere, right? So no energy is required for this, by the way, but it's just called the power stroke. And what actually happens when the power stroke occurs? The head actually releases ADP and inorganic phosphate. Okay, so mainly the sarcomere shortens, or you could say that the Z lines move closer together. So this is the M line, and these are two Z lines. So the distance between the section between two Z lines is called the sarcomere. So when the myosin head, right? This is the myosin filament comes from the M line. These are the heads. So when it actually pulls the actin towards the M line, the Z lines come closer together, right? This is what happens essentially. See that it becomes shorter. No energy is required for this, but we do release the ADP and inorganic phosphate to do so. Or you could just talk about the rowing motion of the head, or the power stroke that works, the power stroke. So what is the function of ATP then? Where is it used? So the ATP binds to the myosin head, right? After the power stroke occurs. So the myosin head actually has an ATP enzyme. So the ATP is hydrolyzed. It is hydrolyzed by ATPase, or the myosin head actually. Now the head actually detaches from actin and it comes back to its original position. The head comes back, or tilts, okay, back to original position. So do you guys understand that the ATP is actually not used for the power stroke? So ATP is actually used to release the myosin head from the actin so that it can come back to its original position. Okay. During contraction, muscles use up ATP very quickly. For a short period of time, ATP can be resynthesized using creatine phosphate as shown in figure 5.1. The creatine formed as a result of the resynthesis of ATP is converted to creatinine. Creatinine production in the body stays fairly constant. Creatinine becomes part of the glomerular filtrate during ultrafiltration in the kidney nephrons. Ultrafiltration requires a high blood pressure in the glomerulus. Explain how this high blood pressure is achieved. Mainly how the afferent arteriole is wider than the efferent arteriole. Or you could say that the efferent arteriole is more narrow. So this actually leads to the high pressure, okay? Since after the glomerulus, when you go from a wider diameter to a lower one, high pressure will be created. And the main filtration barrier in the nephron that allows creatine to pass into the Bowman's capsule but stops the RBC from passing through. So there are three sections, right? Starting from the capillary side, glomerular side, the fenestrations in the capillary, then followed by the basement membrane, then the podocytes. So mainly, the main filter is the basement membrane. All right, moving on. The concentration of creatinine in blood largely depends on the glomerular filtration rate GFR. By measuring the concentration of creatinine in the blood, the GFR can therefore be estimated. The value of the GFR can be used to assess the efficiency of the kidneys. In humans, a normal value of the GFR is 100 cm cube per minute, out of which 99 cm cube is absorbed per minute, by the way. So only 1 cm cube of urine is produced per minute typically. So figure 5.2 shows the relationship between the GFR and the concentration of creatinine in the blood. Describe the relationship shown in figure 5.2. So the greater the GFR, right, the lower the concentration of creatinine. Inversely proportional. So the higher the creatinine concentration, the lower the GFR. Or you could say the other way around, just like I said a bit ago. So you could talk about another point. You could just say that it's an exponential curve, or you could say that it is inversely proportional, or you could talk about the data code for 2 points, right? For example, here and here. So you could talk about at a GFR of 125, there's a concentration of 0.01 gram per dm cube in blood, cm cube per minute. And what about here? This is 10, 20, wait, 1, 2, 3, 4, 10, 2, 3, 4, 20. So this is 10, right? So that's 3 boxes. So at 7.5, right? At 7.5 centimeter cube per minute, there's a concentration of 0.14 gram per dm cube. 0.14 gram per dm cube, right? In the blood. So yeah, that's an option. Or there is another option. There's a hack. You could just go for creatinine concentration is inversely proportional to the GFR. This scores you 2 marks actually. So use figure 5.2 to estimate the concentration of creatinine in the blood that indicates a normal GFR. So we're looking for 100 GFR. So they're talking about this point over here. So this is 0.01, right? So 0.02, in between 0.012 and 0.014. So it's going to be 0.013. Okay, 0.013 and 1 mark for the unit of gram per dm cube. So there's 2 reasons why the GFR of a person may decrease. When does GFR actually decrease? Think about it. Mainly due to kidney disease. Or any damage, kidney disease or damage. It may be due to cancer, it may be due to dehydration, your body wants to retain more water, or it may be due to low blood pressure. low blood pressure, or it may be due to loss of blood, due to hemorrhage. Okay, moving on. So we have 10 questions in total. We're at question number 6 now. The last questions are a bit smaller though. The black pigment melanin contributes to hair, skin, and eye color. Melanin is produced by cells known as melanocytes. Tyrosinase is an enzyme involved in the production of melanin. A study was carried out to investigate the effect of an extract of the starfish on the activity of tyrosinase. It shows the table shows the results of the study. Suggest how the starfish extracts affects the activity of tyrosinase. As the concentration increases, activity clearly decreases in all steps. So the greater the concentration of extract, the lower the activity of tyrosinase. All right. Why though? Actually, what is the extract doing? The extract acts as an inhibitor, or you could say that the enzyme is inhibited. How? The extract binds to active site. It could be acting as a competitive inhibitor or to the allosteric site. Do you remember this? It's basically a site other than the active site which causes a change in shape of the enzyme of tyrosinase, right? Now, it may alter pH, or the extract changes the active site, the shape of the active site actually would be more exact. Or you could just refer to it being denatured. Denatures the active site, okay? Denatures the enzyme. That's typical what a non-competitive inhibitor does, right? So the dominant allele of the tyrosinase gene codes for the enzyme tyrosinase, TYR gene. In people with albinism, the melanocytes do not produce melanin. This is caused by a recessive allele of the TYR gene. What is meant by recessive and what is meant by allele? So basically, what is an allele? An allele is a different form or variety of a gene. Okay. And what does recessive mean? A recessive allele is only expressed when a dominant allele is not present. What does that mean? It is only expressed in a homozygote. Okay. Or you could say that it is not expressed in a heterozygote. Okay. Remember that. Construct a genetic diagram to show how a man and a woman who both produce melanin could have a child with albinism. Okay, that means they may be heterozygotes, right? So let's start. So you need to explain the symbols, right? So A, this is actually an allele for melanin production, or you could just go for the normal pigment, right? What about A? Since it's a recessive disorder, small a is allele for no melanin production, or we could just go for albinism. Okay, so now we mention the parental genotypes. So parental genotypes followed by gametes. So the parental genotypes are capital A, small a, capital A, small a, and the gametes are going to be capital A. You need to circle the gametes, okay? Remember that gametes should be circled. Um, wait. Now let's go for the offspring genotypes, right? Where this is gonna be. We used a Punnett square. I end up with this, right? So this is our child with albinism. Okay, you just need to identify that. This is how it's possible. The other ones are normal, normal, normal. Okay, so that's all. So moving on to 7. Figure 7.1 shows a red deer. Red deer feed on a wide range of plants. The number of red deer in the UK increased from 135k in 1960 to 360k in 2010. Environmental factors affect the population size of red deer so that numbers do not continue to increase. Suggest environmental factors that may prevent further increase in the size of red deer populations. Okay, so predation is a major factor. Then there may be competition for food or a decrease in food available or limited amount of food. There may be a disease or loss of habitat or breeding sites. Or you could say that the habitats are limited, right? It may be due to some pesticides or herbicides, right? Or you could just refer to a change in climate, which may lead to a reduction in the size of red deer populations. Okay, so this is the body mass of red deer. It shows wide variation within a population. Medium body mass is selected for clearly. A selection pressure acted consistently over many years against red deer of low body mass. Okay, so sketch the population after this time. So the selection pressure actually acted here on deer of low body mass. So what's going to happen? Directional selection will occur. Low will disappear after some time. Okay, low will disappear after some time. And the peak will no longer be at medium. It will be slightly to the right. So it's going to be like this. So it was over here, right? Now it's going to go here. It will be something like this. Okay, so the same peak, right? Same shape, more or less, but to the right. There was an alternate way to do this. You could also go for this one. This also works. This is also okay. Name the type of force of natural selection. Okay, so this is actually directional. This is directional selection when one extreme is selected against. What about this? Now, against medium body. So if it acts at the middle of the curve, disruptive selection will occur. Now we end up with two peaks, right? With a dip in the middle. So this is called disruptive selection. Really important diagrams. Okay, so the position does not change. It's kind of like this, yeah. Now, during a sporting event, an athlete carries out aerobic respiration. Structures and compounds involved in aerobic respiration are listed from 1 to 10. So, these are 10 compounds. Complete table 8.1 by matching each description with one number chosen from 1 to 10 to show the correct structure or compound. You may use each number once, more than once, or not at all. So location of ATP synthase, where is it located? We know that it's a stalked particle located at the inner membrane of the mitochondria, okay? The cristae basically, so it's going to be the number 7. Transports hydrogen atoms. Basically a hydrogen carrier, either NAD or FAD. We needed a coenzyme, so number 4. Nucleotide with a purine base. Okay guys, I'm going to teach you about nucleotides. Nucleotides are of two types. Purines or pyrimidines? Purines are the double ring structures, adenine and guanine, and pyrimidines are the single ring structures, uracil, cytosine, and thymine. So the purines are adenine and guanine. So we need nucleotides with a purine base. So mainly it's ADP, adenosine diphosphate, right? And NAD also works. NAD also works. What is NAD? Nicotinamide adenine dinucleotide, right? That is why it also works. So adenine and guanine. These are the two. Location of substrate linked phosphorylation. So this mainly occurs where? In glycolysis and in Krebs cycle. So where does glycolysis occur? It occurs at the in the cytoplasm. So number 2. Number 2 is the best answer. Enters the Krebs cycle. So this actually enters the Krebs cycle from which part? Like followed by glycolysis, we have the link reaction where we get acetyl CoA. So that acetyl group actually enters the Krebs cycle. So number 10. Processed by oxidation of triose phosphate, pyruvate. Okay, this is in glycolysis. So pyruvate is oxidized to form triose phosphate. Sorry, triose phosphate is oxidized to form pyruvate. We get 2 molecules of pyruvate from 1 glucose molecule. These are the steps of glycolysis. You should know the basics. So sometimes the muscle cells of an athlete need to carry out respiration in anaerobic conditions. Explain why the respiration of glucose in anaerobic conditions produces less ATP than in aerobic conditions. Very, very important. Okay, let's go. So mainly what occurs? Only glycolysis or conversion of glucose into pyruvate occurs. So this only produces 2 molecules of ATP. So this is the net production. Okay, basically we get 4 molecules of ATP, but the net production is 2 because 4 minus 2, we input 2 molecules of ATP, right? So we get 4 but input 2, so 4 minus 2 is 2. So it only produces 2 molecules of ATP net, okay, per molecule of glucose. Why is that? Because only substrate linked phosphorylation occurs. Don't use the term substrate level phosphorylation, by the way. According to the new syllabus, it's substrate linked phosphorylation. So what happens afterwards? Pyruvate is actually converted to lactate, and remember, lactate is still energy rich. That means all the energy from glucose has not been broken down yet. That's why we get less ATP. Some of it is still stored as chemical energy within lactate. Why is that though? Because oxygen is not available, right? Oxygen is not available as the final electron acceptor in the electron transport chain. So the electron transport chain or chemiosmosis or oxidative phosphorylation does not occur, and most ATP is produced in aerobic conditions in the electron transport chain, or chemiosmosis or oxidative phosphorylation, right? Also, no additional substrate linked phosphorylation from Krebs cycle. So hear me out, this point is quite interesting. Typically, more substrate linked phosphorylation is supposed to occur in the Krebs cycle, but think about it. That pyruvate that we're getting from the from glycolysis is not transferred at all to the next step because it's converted to lactate. Or else think about it, all the NAD would become reduced NAD. So to get NAD reduced NAD oxidized to NAD again, we're actually sacrificing this pyruvate. So since it can't be taken to the next step, that is link reaction, no acetyl group is entering the Krebs cycle, so no substrate link phosphorylation is occurring there as well. That's the problem. That's why we get so less ATP, only net 2. Now, the diversity of dung beetle species was investigated at 2 grassland sites in North America. Dung beetles feed on animal feces dung, right? So the first grassland site was grazed by cattle, and the second grassland site was not grazed by cattle. The areas of the 2 grassland sites were the same. At each grassland site, dung beetles were collected, identified, and counted. Okay, so we're using Simpson's index of biodiversity here for the dung beetles on the grassland site. Grazed by cattle was calculated as 0.522 using this formula. So we need to complete figure table 9.2. Right. So what does capital N represent? It represents the total number of species, individuals, and small n represents the individuals of each species present in the sample. So let's start. So we're going to find this one for the site that is not grazed by cattle, this one. So let's find out the total. It's 7608. So for A, it's going to be 6641 divided by 7608. 7608. It's 0.87289. Oh, we need to show our working. Okay. So 6641 divided by 7608. 0.3 decimal places, right? 0.873. What about the next one? 774. 774 by 7608. This is 0.102. Next one, 85. 108 and 85. Okay. Zero point 0.0142. 0.01117. So to 3 decimal places, this is 0.014. This is 0.011. Okay. Wait, we shouldn't do it this way. My bad. Just round it off directly. So I'm getting 0.01117. So it's just going to be 0.011. What about that one? 108 by 7608. That's going to be 0.014. Okay, so that's it. Okay. So let's fill the table up. 0.873, 0.102, 0.014, and 0.011. Now we square these, right? So it's 0.762. Then 0.762. 0.1035, so it's 0.010. I'm doing the last one first. This square, right? This square is 1.248 into 10 to the power minus 4. So that's 0.000124.8, right? So this is 0.000. What about that one? 108 divided by 7608 whole square is also 0.00020151, which is also 0.000. So if we add all of these up, what are we getting? I'm getting 0.772, right? So what's the value of capital D? It's 1 minus the summation. So 1 minus 0.772 is 0.228. So this is how it works basically. The closer the value is to 1, the more the biodiversity. So it's very low, right? It's 0.228. So it's not that diverse. So let's compare this. This is the one for not being grazed, and this one was the one that is grazed by cattle, 0.552. So clearly this one has more biodiversity, okay? So let's give the reasoning. So we can say that there is a greater species evenness on grazed grassland. Or you could say that there's mostly only one species, right? This one. Okay. Anyway, what else? So we can reach a very conclusion that grazing actually increases dung beetle species biodiversity. Okay, this is our conclusion. Okay, so let's go to the other one. How to actually find out or estimate the population size of a species of beetle that does not feed on dung in each of these two areas. Okay, so mainly beetles are mobile species, right? So random sampling won't work. We specifically need to use the mark release recapture technique. There's one mark for mentioning that. Now, how do you get the other three? So mainly you gotta give some details about trapping them, a detail on marking them, right? So we can use a pitfall trap, right? A pitfall trap can be used to trap the beetles. Then we can use a felt tip pen, or you could talk about marking the beetle in such a way that it does not have adverse effects, so that it doesn't become easier for predators to spot, right? You could say that. Then we will release the marked beetles. All right. Then we will release the marked beetles, and then we will catch some beetles a second time. Now, when though, it should not be too soon. Like it shouldn't be after 1 hour of catching them the first time. After catching them the first time, because there won't be enough time for mixing, and it should not be too long, right? It should not be after 1 or 2 weeks. Then migration may have occurred. So that's unfair, right? We won't get enough beetles. And then we can give the Lincoln index. This is how we find out the number of beetles that we captured. So the number of beetles is equal to the number of marked individuals released. That means we're talking about the first time multiplied by the number of individuals captured in the second time, right? This includes both marked and unmarked individuals. Divided by the number of individuals recaptured the second time that are marked. Okay, this is also the second time, but only the marked individuals. Okay, so that's all. So this is very important for the Majun series, I think, and important for paper 5 as well. Now we're on to the last question, which is explain genetic control of protein production in a prokaryote using the lac operon. Okay, so let's begin. So mainly the lac operon consists of structural genes and regulatory genes. So the regulatory gene codes for a repressor protein. So what does the repressor do? The repressor protein binds to the operator on the gene. So this is an example of an inducible enzyme. The enzyme beta galactosidase will only be produced when the substrate lactose is present. Okay, so in presence of lactose or allolactose, right? What happens? Lactose actually binds to the repressor protein. So this was the main setup, right? This was the setup. So we have the structural genes Z, Y, and A over here, and we also have the promoter here. This is a promoter, and we have the regulatory gene. This is the regulatory gene back here, which codes for repressor protein. So we also have an operator section here, right? The operator section. So the repressor protein, right? This one, I mean the regulatory gene, it actually codes for a repressor protein which binds to the operator region. As a result, the RNA polymerase cannot bind to the promoter because the repressor protein is blocking the way. This repressor is blocking the way. So when lactose comes over here, what actually happens? Lactose actually breaks down this repressor protein or binds to the, you know, it actually changes the shape of the repressor protein after binding to it. Okay, so after lactose binds to the repressor protein, it changes shape basically, right? And moves away from the operator. Okay, now what? Now you can say that promoter region is now unblocked, right? Now what? RNA polymerase can now bind to promoter, and ZY genes, the structural genes, are now transcribed, or you could say that they are switched on, right? Or the mRNA is now synthesized. So enzymes, or you could talk about beta galactosidase, a named enzyme, can now be synthesized. So you had an option. You could also go for I answered this question in a way that when lactose is present, what actually happens after binding to the repressor. You could word it in this way. You could say that if lactose was not present, what would happen? The repressor protein actually covers part of the promoter, so RNA polymers cannot bind to it, and the structural genes cannot be transcribed, and the enzyme can't be synthesized, so lactose is not broken down. Okay, so that's it for the specimen paper. Very important for the um, Major series. If you have other questions, you can ask me. And I'm going to link the February March paper over here, right? And remember to subscribe to the channel. Okay, see ya.